All right, gang, we've made it. We've made it through those simple chemical senses, through the barbaric mechanical senses. What even is that? Just stuff bonking into other stuff? Come on with that. Now we've made it to the crown jewel of the human sensory system, vision. I feel like a tour guide at a museum where I've been showing you all the quiet wings of the museum, like the vestibular system and the olfactory system and the well-respected but less popular exhibits like the auditory system. And now I'm taking you to see the Mona Lisa or the Rosetta Stone. For humans, vision is the sense that we hold in esteem over all our other senses. You can even see the importance of vision reflected in language. Language is full of phrases that use seeing synonymously with understanding. Do you see what I mean? It's also the sense that people report being the most reluctant to part with. Rhinos wouldn't want to lose their sense of smell. They've got a great one. Lobsters wouldn't want to lose their somatosensory system. They've got an incredibly sensitive somatosensory system. For us, it's our eyes. There's more research on vision than any other sensory system. And so in part because of that, and in part because it's so important to us, we'll spend more time on vision than any of the other senses that we've talked about so far. So today we're going to start off with the anatomy of the eye and the process of visual transduction, and then in future lectures we'll talk about color, depth perception, motion processing, object recognition, and face perception. We're not going to be talking much about multisensory integration in this unit because we've already talked about how vision is integrated with all of our other sensory systems. So to begin with, as you read, the stimulus for the visual system is of course light. And it's not all light. It's a very narrow band of the electromagnetic spectrum called the visible spectrum that ranges from about 400 to 700 nanometers, that ranges from wavelengths of light about 400 to 700 nanometers long. The first step towards visual transduction begins with these beautiful windows to the soul. The iris, the colored part of the eye, is not involved in visual transduction, but people are always interested in this. So I'll tell you that what determines eye color does not have to do with different colors of pigments within the iris. That is, the only coloration that an iris can actually have in it is a kind of brownish pigment called melanin that exists in the stroma, which is the layer right at the back of the iris. People who have brown eyes have a lot of that pigment, and so when light contacts those pigments, it's mostly absorbed, and just the little bit that's reflected to us looks brown. People who have blue and green eyes, on the other hand, don't have greenish and bluish pigment in there. Instead, they have lower levels of melanin and different amounts of collagen deposits in the eye. So light ends up being primarily reflected and scattered in what's called the Tyndall effect. That's actually somewhat similar to Rayleigh scattering, which is what makes the sky blue. The really interesting part of the visible external eye, though, is the pupil. For humans, it's round, but you may have noticed that for other animals, it's different shapes. So for instance, cats and other animals that are active in the day and the night have pupils that are shaped like vertical slits because that shape allows the pupil to change size much faster than the round pupils that humans have. So the smaller the pupil, the less light can come in. And so cats are much less likely to get blinded by sudden changes in light level than humans are because those slits can close so rapidly. So species with those kind of vertically elongated pupils are very likely to be ambush predators that are active at both day and night. Well, the first thing to point out is that light enters the eye through the pupil, that black part right in the middle. Except, get this, it isn't actually a black part. The same way that you wouldn't look into a well that you can't see the bottom of and say, that well is black colored, there isn't actually anything that's black in your pupil. It's just a hole that leads into the middle of your eyeball. So there's a lens and a cornea that cover it, so you couldn't just poke your finger into that hole. But if the lens and the cornea weren't there, you could. The pupil is just a hole. So the next time you're staring someone deep in the eyes, keep in mind that what you're actually seeing is the blackness inside of their eyeball. How cool is that? Okay, so our stimulus is light. Our next goal is figuring out how to get the information to focus right on the fovea rather than in front of it or behind it. So the process by which we do this is referred to as accommodation. All right, so here's an example. When an object is far away and the eye is relaxed, the focal point of that visual stimulus lands directly on the fovea, and we can transduce it and see it. But if the object gets moved closer to us, the image no longer falls directly on the fovea. That focal point now falls behind the fovea, behind the retina. And so in order for us to move that focal point onto the retina, we need to contract the ciliary muscles, which leads the lens to thicken and to the light to become refracted differently such that it now lands on the retina. So you can feel yourself do this, right? As you move your finger closer and you keep it in focus, you feel that kind of like pull and tug in your eyeballs, right? You feel that little bit of burn. So those are the ciliary muscles tightening up, making it so that that focal point falls directly on the retina. Let's watch a little video of how this works. So look at those little ciliary muscles. As they're contracting, they change the shape of the lens so that as light passes through it, it bends, it refracts a little bit differently and falls on a different point. Now, as we age, the eye changes in several important ways. The first is that our near point, that is the closest point at which we can comfortably accommodate to, gets farther away. So there gets to be a point where things get so close, you can't focus on them. So for me, how far is this? I can't focus on this. I'm squeezing my ciliary muscles as hard as I can, but I just can't focus on that. And so that the closest point at which we can focus gets farther away as we age. This happens in part because of hardening of the lens in the eye and also of weakening of the ciliary muscles. So many older people wear reading glasses because they want to be able to see things that are close enough that are closer to them than their near point, closer to them than they can accommodate to. The most common causes of imperfections in vision are myopia and hyperopia, as you read about. And these can occur primarily because of deformation of the eyeball or cornea. In order for the eyeball to work perfectly, in order for you to have 20/20 vision, the eye has to be nearly a perfect sphere. So any deviations in the shape from that can lead the focal point to fall too far in front of or behind the fovea. And those differences, the difference in where the focal point is and where the retina is, are too big to be properly accommodated. So we can't resolve them just via accommodation, and so people typically will wear glasses or contacts in order to bring the focal point to the fovea. So we can correct for vision using glasses and contacts that change where the focal point falls so it is within the range that we can sufficiently accommodate to. Another way that we can correct for vision is using what's called laser eye surgery or Lasik. So the way that Lasik works is to reshape the cornea, which is that transparent covering at the front of the eye. Although the mechanism is different, the idea is similar to glasses and contacts. The goal is making the focal point fall closer to the fovea so that the eye can then accommodate and do the rest of the work to get the focal point directly on the retina. So the first challenge of the visual system is getting light to fall in the right spot. Another challenge is getting there to be the right amount of light. So one way that we can adapt to differing levels of illumination is for pupil size to change. So for instance, when you're in a dark room, the pupils dilate, they get much larger, which allows more of those low levels of light to get in. If you're outside and it's really bright, the pupil rapidly constricts to allow less light in to avoid the visual transduction system from being totally overwhelmed by that bright light. Okay, so we've got our stimulus. It's a narrow range of the electromagnetic spectrum. We've got a way for light to get to the light-sensitive tissue in the retina. It's via accommodation. And we've got a way to control the amount of light that gets in. It's pupil size. So now we need a way to turn that light into action potentials. All right, so assuming that the shape of our eyes and the distance to the object and the focusing power of the lens have conspired to make the focal point of the image fall on the fovea, and the pupil is appropriately constricted, so we've got the appropriate level of light, how do we deal with that stimulus? Well, at the back of the eye is the light-sensitive retina. And as you can see, there are many layers of neurons there waiting to respond to that light. Here's a more zoomed-in view. You can see that there are different types of neurons here. I want to really emphasize the unintuitive layout of the retina. So in this image, light is coming from the left side. So the front of the eye is on the left side. But the photoreceptors, the light-sensitive neurons, are at the very back on the far right side. So that means when transduction happens in the photoreceptors, that information is then passed from the photoreceptors back toward the front of the eye again. So when light falls on a given photoreceptor, it causes a chain of events that ultimately result in action potentials in the retinal ganglion cells shown in yellow here, and those signals then being carried up to the brain. So why this counterintuitive layout? Why not have the photoreceptors right up at the front so that they don't have to pass through layers of other neurons in order to be transduced? It's not entirely clear, but it may, that this layout may selectively filter certain frequencies of light, which is beneficial for color vision during the daytime. It may also help to regenerate the photopigment that are contained within the photoreceptors, because the tips of the photoreceptors are embedded in what's called the choroid, which is, you can see in the red on the very right side of the screen, which has a rich blood supply, and so it can easily get nutrients to those photoreceptors. It's important to point out that even though we talk about the retina as being this one light-sensitive organ, the retina is very different depending on where in the retina we're examining. So if the inside of the eye were well lit and you could look in through that black hole of the pupil, this is what you would see. So at the center, the fovea, that's where images fall on the retina if you're looking directly at them. In the fovea, the retina contains only cones. Out in the periphery, in peripheral vision, it's a mix. It's mostly rods, but there are also some cones there as well. A few years ago, I went to the eye doctor and they were doing imaging of my retina that looked like this. And I chatted up the doctor and I told him, that I teach this class and I'm so interested in eyeballs. And I got him to send me digital versions of my eyeball pictures. And so I usually in this class show my own retina here, but this one isn't mine. And here's why. Everyone's retina is different. The image is unique to a person based on the particular arrangement of blood vessels. It's like an eyeball fingerprint. So you can use retinal images as identification. Sometimes they do this in movies. You see like a retinal scan. The science behind that is actually pretty sound. So I don't put pictures of my own pretty retinas on the internet because I don't want to get identity thieved by Tom Cruise trying to break into a secret plant in Mission Impossible or something. So this is just some internet person's retina. Good luck, Tom Cruise. Okay. To be honest, it looks a lot like mine. I don't know, mine's cuter. Okay, so here's a representation of that same thing in graph form. At the fovea, there's lots of cones. Out in peripheral vision, there's more rods, but a few cones elsewhere as well. And there's a big chunk of the retina that has no photoreceptors, and that is the blind spot. So because of the particular kind of backwards arrangement of photoreceptors and retinal ganglion cells, there has to be a place in the eyeball where the axons of the retinal ganglion cells that make up the optic nerve, there has to be a place where those exit the eyeball. And in that area, there are no photoreceptors. So that means that any light that falls on that region of the retina can't get transduced. There are no photoreceptors there to pick up on it. So in the image that I just showed you, the blind spot is where all of those blood vessels kind of collect together. That's also where the blood vessels leave the eye. So there is a chunk of the retina that you can't see anything in that happens kind of out here in peripheral vision. All right, so now that we have gotten light to land on the photosensitive tissue at the back of the eye, now we need some way of turning that light into action potentials. So in every sensory system, we've gone from some kind of physical energy or stimulus, and we've ended up with action potentials leaving that sensory organ, traveling up to the brain via the relevant sensory nerves. You may have noticed that our processes of sensory transduction have been getting more and more complicated, right? We start with just salt entering ion channels, super straightforward, then onto the more complex forms of mechanical transduction, and now we're up to the most bananas kind of transduction that there is. The process of visual transduction is so complex that we're actually going to kind of skim through and gloss over several parts of it. So rather than having you focus your attention on the very complicated cellular mechanics, I want us to have plenty of time to get to the higher order perceptual processes. So when I walk you through the process of visual transduction, I'm going to do it in two parts. The information on the left is the content that you are responsible for. This is kind of the summary of the process of transduction. The stuff on the right is bonus content. That means you're not responsible for it. It won't show up on the exam guide, but I'm going to include it because it might help you get an understanding of the process. So visual transduction begins when light contacts a pigment in the photoreceptors called retinol. So here's a schematic of a rod. Inside the rod are discs that contain the visual pigment molecules that have two components. So opsin are these large protein strands, and retinol is the light-sensitive molecule within. And visual transduction happens when the retinol absorbs a single photon of light. So when the retinol absorbs a photon, it causes one of the double bonds between the carbon atoms to break down, and that makes the retinol change its shape. The process of the retinol changing shape we call isomerization. Now, what's really wild about visual transduction is that in the dark, in the absence of a light stimulus, sodium channels in the photoreceptors are normally open, meaning that in the dark, the cell is depolarized and releasing neurotransmitter glutamate. Yeah, gang, you heard me right. In the absence of a stimulus, the cells are releasing neurotransmitter. So the process of isomerization, when that retinol changes shape, that closes the sodium channels, which leads the cell to hyperpolarize. So the isomerization causes the cell to be activated, but not in the traditional sense that we talk about cells being activated where they're depolarized and starting to release neurotransmitters. No. In this case, they're hyperpolarized and they stop releasing neurotransmitters. So the signal that the later neurons are picking up on is, hey, that cell stopped releasing neurotransmitters, rather than, wow, I'm excited by the start of neurotransmitter release. Okay, so the isomerization closes the ion channels, which causes the cell to hyperpolarize. So this hyperpolarization leads to lower levels of glutamate being released by the photoreceptor. And the amount of neurotransmitter released by each photoreceptor is influenced by the horizontal cells that can modulate the photoreceptor response. The changes in neurotransmitter release depolarize the bipolar cells. There are, in fact, also bipolar cells that hyperpolarize in response to the reduced neurotransmitter release, but we're not going to deal with those. Okay. So depolarization at the bipolar level leads to depolarization at the level of the retinal ganglion cells. So at this point, we're back to a system that makes sense. We've got depolarization in the bipolar cells leading to depolarization in the retinal ganglion cells. And once the retinal ganglion cells have depolarized, that's really the end of the tricky part. So the depolarization in the retinal ganglion cells become the action potentials that are sent up the axons of the retinal ganglion cell, which make up the optic nerve and up to the brain. Because this is interesting, there are also some kinds of retinal ganglion cells that hyperpolarize in response to activity at the bipolar level, but we're not going to talk about those as well. All right, so to summarize, the key points that you should have a good handle on that enable us to understand the higher level perceptual stuff is that light makes contact with the photoreceptors. That causes the retinol within the photoreceptors to isomerize or change its shape, which leads to an electrochemical cascade that ultimately results in retinal ganglion cells depolarizing, firing action potentials, and those action potentials are then carried up to the brain. All right, it's a short lecture today. I wanted to make sure you have plenty of time to go back and rewatch and review your notes to make sure you have a good handle on these processes that we've talked about and the nature of light before you start in on the problem set in class. I'll see you later. All right, here is a schematic of you using the blind spot finder. If we could just look at your eyeball and the inside of your eyeball as you're doing this, here's what we're going to see. So the information about the cross is landing right on the fovea of your eye. The information about the circle, when the blind spot finder is positioned correctly, that information is landing on the chunk of the retina that doesn't have any photoreceptors in it because it's where the axons are leaving the eye. Now, it's often very hard to find the blind spot because you have to get everything set up just perfectly. If, for instance, you were a little bit too far away from the blind spot finder, look what would happen. Now your eye is scooted back a couple of inches, and now that cross is still in foveal vision, but the circle is now falling on a point in the retina that has photoreceptors. So if you weren't able to find your blind spot, it's probably because you couldn't get the circle lined up perfectly so that it was falling right on the blind spot. So if we actually have these like big, pretty sizable holes out in vision, why don't we notice big blind spots, right? Why don't I just have like big black holes in my vision out here? So there's a few reasons. One is that the region of the visual field that the blind spot falls in is covered by the other eye. So if my blind spot is like right here, if I close this eye, I can still see that point with my right eye. So you could only conceivably ever see your blind spot when one eye is closed. The other reason is that the blind spot is in peripheral vision, far out in vision, which has relatively poor acuity. If you had a blind spot sized region in foveal vision right at the center, it would be super obvious to notice. But our vision out here is pretty bad anyway. Another reason is that we move our eyes a lot. Even if you're not consciously aware of it, we're constantly making small motions. And so information isn't likely to be in the blind spot for very long because we move our eyes so much. Another reason is that we fill in the missing information using top-down information. So if you, for instance, do a blind spot demo where rather than just having a circle on a white background, you have something that's, a complex pattern like a plaid or a checked pattern or something, we still tend to see that complex pattern even though we have a hole in vision. So it's not like we're just replacing the hole in vision with, white. We're replacing it with what's immediately around it. So with photo red eye, this happens when people are in dark rooms and their pupils are dilated, so they're open wide. When the light of a camera flash happens, the light is reflecting off the blood vessels in the retina. So that red picture that I showed you earlier, you're basically getting a flash of that shown through somebody's pupil. Remember, these are like, the windows to the retina. They're not the windows to the soul. And so if you're in low light and you have a big flash of light, you're seeing their blood. The pre-flash flash is useful for reducing red eye because the pre-flash flash makes the pupils constrict and get smaller, and then it's more unlikely that the light that goes in from the flash is going to reflect out in just the same way and therefore get picked up on by the camera. Cool, right? All right, so we go from having over 100 million photoreceptors to having 1.5 million retinal ganglion cells. And what that means is that we've got a lot of convergence as we go from photoreceptors to retinal ganglion cells. There's more convergence out in peripheral vision than there is in foveal vision. So in foveal vision, there may be even a one-to-one relationship between photoreceptors and retinal ganglion cells. So you may have like a single cone that is sending messages to a single retinal ganglion cell. Whereas out in foveal vision, you're going to have lots and lots of photoreceptors that are all summating on one retinal ganglion cell. So when you have more convergence, that means you're going to have larger receptive fields, right? Out in peripheral vision, you may have a spot this big, and all of the neurons that code for that spot are connected to a single retinal ganglion cell. Whereas in foveal vision, you're going to have just a little spot that makes up the receptive field. So if there's more convergence and larger receptive field, there is less cortical representation. So there's a really nice parallel to be made here with the somatosensory system, where foveal vision is like fingertips and peripheral vision is like the back. The similarities with the somatosensory system don't end there, because there are different amounts of cortical representation related to which areas of the visual field are represented. So for instance, foveal vision, so that's what's shown in orange here, even though it makes up a relatively small proportion of the visual field, has a really large representation in cortex. So we've got a big chunk of our occipital lobe of V1, primary visual cortex, that's devoted to coding for foveal vision. All right, so in this great adaptation example, the light is falling on the light-sensitive retina, and if a particular photoreceptor is receiving exactly the same input, its response is reduced. We're going to talk more about adaptation in the visual system and the consequences that this can have for color perception in a couple of class periods. One of my least favorite facts that I ever learned is that the plural of octopus is not actually octopi, it's octopuses. Because I used to make a great joke about octopi, but I can't do that anymore because it's not technically correct. But how funny would that have been? All right, so the deal is that octopuses don't have a blind spot because all of their photoreceptors point forward, point toward the light source. That means that the retinal ganglion cells that they synapse on just come off the back of them, get tied up into a nice bundle, and go up the optic nerve. So they don't have that point where the retinal ganglion cells have to pass through the photoreceptor layer. Why are octopus and squid eyes arranged in this fashion? Again, it's not entirely clear. It may have to do with the fact that there were different evolutionary demands on the kind of light that could be filtered by the eye, given that they see underwater and in lower light situations than humans do. It may also be they don't need to regenerate photopigment as quickly because they see underwater, and so the pigment isomerizes or bleaches less. It's not entirely clear. All right, this one's creepy. Sorry. If you could actually look through someone's pupils, you would see something like this in their retina. So the image that you got was the right eye, so on the left side of the screen here as you're facing the person. So if you look inside their eyes in front of them, the fovea is always more peripheral, it's always more on the outside than the optic nerve. The optic nerves kind of point in, and the fovea is going to be on the outside of them. So that's why the blind spot is in the visual field So that's why the blind spot always falls in peripheral vision away from the nose. So now we have a way of getting the appropriate amount of light into the eye, getting it to fall in exactly the right place so that it makes contact with photoreceptors, and a way to transduce it to send those signals up to the brain. Now you might think that the visual system then is just as simple as saying, well, how much light is present and what wavelengths and so forth. I just want to give a little bit of a teaser going into next time that even though we have talked so far and focused on the way that we convert light into action potentials, there's still ways that we interpret those action potentials. So there are still top-down influences on how we perceive that light. So for instance, in the cube here, what color does this appear to be? And what color does this appear to be? Yeah, in fact, guess what? They are in fact exactly the same shade. So in addition to the information that we're getting about the actual light sources that are present, we're also using information about our expectations based on some of the other cues in the picture. So even though we've focused today on transduction and stimulation of photoreceptors, I want you to keep in mind that there's actually a lot more to vision than just stimulation of photoreceptors. We'll talk more about that in the upcoming class periods. I will see you next time.